Good afternoon, everyone. I'm Colin O'Connor, Custom Content Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Exploring the Role of MRD in Oncology Drug Development Toward Precision Diagnosis and Personalized Treatment, and it's sponsored by Burning Rock. Our speakers are Johannes Nipkin, Chief Medical Officer at Ionova, Chi Shea, Chief Medical Officer at Impact Therapeutics, and Zhou Zhang, Chief Technology Officer at Burning Rock. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Johannes Nipkin.
Hello, my name is Johannes Nipkin. I'm an independent consultant for pharmaceutical strategies for pharmaceutical companies, and I was Chief Medical Officer for several companies in China. The last, until recently, was Ionova Life Science in Foshan. And I am here to talk about the clinical applications of MRD dynamics. The evolution of MRD and MRD detection is a quite hot field currently. As we all know, in hematological malignancies, we had already a lot of experience with minimal residual disease. As in all cancers, we would, of course, like to know if after the primary treatment, maybe surgery, radiation, or other treatments, there's still disease in the body. And even if there is a minimal residual amount, we know that this can lead to disease progression and relapse. And so want to find that out. For hematological malignancies, as this is a tumor where the tumor cells are anyway shed in the blood, this was already a good approach, but also lacking a lot of sensitivity. Now with ctDNA detecting methods, we have much better tools, much more sensitivity, and we can see if there's patients who can still harbor minimal residual disease after their primary treatment. The most interesting thing is that with these new methods, where the detection level is much better and much lower, we can also add that to the treatment of solid tumors. And several publications showed that the sensitivity of these assays must be really high, like smaller than 0.01%, to be really able to not have too many false negative results. If this is in the end the outcome, if this is in the end also what we want to see, and if this will be the cut point, the cutoff, where we are saying this patient still needs treatment or this patient does not need treatment, this is of course a different topic. This is something that in clinical studies has to be defined. But for the detection, we can say the smaller, the better. To enhance, to improve these assays, there's currently several things ongoing regarding methods. One is about assay enhancements, increasing input, increasing ligation efficiency, increasing depth, increasing the detected sites, but also noise reduction with different approaches, as well as the capability expansion. What is currently available? We have basically two different kinds of assays. The one are tumor-informed, and the other are, as they are currently called, tumor-naive. I still prefer the term tumor-agnostic, where we have, for example, fixed panels that could be tumor-informed, but could also be tumor-agnostic. And this is the CAPSEC panels. And then we have the purely tumor-agnostic assays that also include methylation detection with the Guardant Reveal and the Guardant Infinity. We have, in this regard, personalized panel that are tumor-informed, of course, not only tumor-informed, but also patient-informed in many cases. So this is the amplicon-based, the Signatera and the PCM. And also hybridization-based, which is BR Prophet and NeXT Personal. Additionally, here we can also face ED sequencing. Finally, We can do whole genome sequencing with tumor-informed approaches. And here we have the MR Detect that is on the market. For the clinical utilities of MRD detection, we are coming back to this, to the old scheme of tumor kinetics, where we, in this case, not only have the limit of imaging detection, but we also included the limit of ctDNA detection. And we can see that, of course, the ctDNA detection can be done on a much lower level than the detection by imaging. So, and we have a certain lead time from particularly the clinical diagnosis than the primary therapy, like surgery, where we before could only see the tumors when there was already significant tumor mass around. But now, with this works with minimal residual disease and the limit of ctDNA detection, we can define and find these patients much earlier. The question is, does it make sense? And here we have, I want to present two studies that were recently done. One study is the EBLIS study that was done in patients with breast cancer. I show the flow chart, but in general, it was around a little bit like 150 patients that were eligible and evaluable from the study. And these were patients with breast cancer after adjuvant treatment. And on this slide, you can see the ctDNA level at any time. And the green part above on the picture A is the patients who were ctDNA negative, and the purple part is the patients who were ctDNA positive. On slide B, the same. This is but for overall survival. And now looking at the next figures, figure C and figure D, we are seeing the same, but based on the patients with a baseline detectable ctDNA level. I have to say the methodology, methodology of the study was a little bit unclear in this regard, because the baseline was defined as the first measurement of ctDNA after the primary diagnosis, and this was done over a broad range of time. So there was no certain time point of this, but still I think the result that we are seeing is quite clear, quite reliable, that basically those patients who had at the beginning or later developed ctDNA positivity had a much worse outcome than those who were very clearly ctDNA negative from the beginning. This is basically kind of seeing a prognostic marker, because in this study, these patients were all treated. But in the next study, which was a study in colorectal cancer, the Galaxy study published in Nature Medicine last year, the study had a quite complicated concept. So it is a very interesting study, but the essence of what I want to show here is about a huge number of patients who were or were not treatment with adjuvant treatment after colorectal cancer. And when you are looking at all stages, we are seeing that the patients in blue who were ctDNA negative had a very low rate of relapsing patients, and the patients who were ctDNA positive had the usual outcome after this treatment or non-treatment. And looking at the first plot, we are also here clearly seeing that basically the only really significant prognostic factor associated with increased ROF recurrence was the ctDNA. All other, usually clinically also used prognostic factors were balanced across the two groups. And here the study was a bit better than the breast cancer study, because the patients had a very clearly defined first ctDNA measurements, which is 4 weeks after surgery. And the patients who were ctDNA negative had basically, which you see in figure B, had no benefit from the adjuvant treatment. But those who were ctDNA positive in figure A had a very clear benefit. And so also a quite clear sign that those patients who were ctDNA negative are in the end also those patients who really have no minimal residual disease. So an adjuvant treatment might not make sense in these patients, at least according to that study. Another interesting finding is that they also looked at patients who converted from their ctDNA level. The dark blue patients are those who were persistently negative in ctDNA. The light blue patients below is the group that converted to positive during the treatment and during the study. And for these patients, you can quite clearly see that they had a little bit worse prognosis. And then when we are seeing the patients who were persistently positive for ctDNA in red and the green curve with the patients who were converted to positivity, then it's also quite clear that patients who became positive, for example, during treatment, had a much better prognosis than those who were persistently there. So looking at the tumor dynamics field again, we can say that the MRD detection can help stratify patients who may benefit from neoadjuvant therapy, can stratify patients who may benefit from adjuvant therapy, and differentiate pseudoprogression with a true progression, and also can support an early implementation of adjuvant or escalated therapy to prevent development of metastatic disease. Also suggest changes in the treatment regimens, so when to change and also how much the change must be. Is this implemented? Is this knowledge that has already been brought into the guidelines? For hematological tumors, where minimal residual disease is a quite well-known and quite long-known principle, it is implemented. So for multiple myeloma and acute lymphoblastic leukemia, as well from diagnosis over induction treatment, over consolidation treatments, or the bone marrow transplantations up to maintenance or monitoring, there are certain time points in the guidelines where an MRD assessment is recommended. So MRD assessment in this case doesn't really define which kind of MRD assessment, but of course the most sensitive is the better in this regard. We also see that there is a lot of studies, namely 82, that have looked at this in hematological malignancies, and only very few studies, like 13, who were looking at that in solid tumors. And this was mainly colorectal and breast cancer, as I just presented before the results. Also for the positive and negative predictive values, median-wise, we can see that this all really makes sense. So that there's a high probability that minimal residual disease assessment with ctDNA, in contrast to our old tumor markers, it also makes sense in solid tumors. And here currently mainly in colorectal, but in the breast cancer, but we of course have to see there might be others. The emerging clinical value of ctMRD assessments also shows a very clear need of standardization. A lot of papers are talking about these, and last but not least, in the NCCN guidelines, it was called that ctDNA as an emerging prognostic marker has to be evaluated, but doesn't make any recommendations how and why and so on. So the need is seen, but there's still a lot of work to be done that we can advance with this. The challenges to advance is the standardization. On the one side, on the testing method, because there's so much around. It is not only about the tumor-informed and the tumor-naive or agnostic assays. It is in general also which kind of genes, which kind of things these assays include. It is about the sensitivity. It is about when we call a detectability in the end minimal residual disease, or when it might be still the noise, or when it might still be something that might not have to be treatment. We don't know about the collection time points for landmark and surveillance. We don't know if it is appropriate for non-shedding tumors. And actually, we don't even know if there are non-shedding tumors, because maybe the more sensitive all these becomes, we will in the end find out that no tumor is basically not shedding circulating tumor cells, and with this also going for the ctDNA. Last but not least, it is about how to define molecular response, how to standardize the references during the validation, how to choose orthogonal methods with or without clinical efficacy analysis. So also with regard to companion diagnostics, for example, and how to establish what is positive and what is negative. And this is probably, as I said before, the most important thing here, because this probably is tumor specific and maybe even stage specific for the different tumors. So you see there's a good opportunity how we can answer this question, how we can come closer to the question if after the primary treatment the patient is really cured or might not be cured and needs additional treatment. But we are not there yet. So there is still a lot to do on the way. But I think all our friends from the diagnostic companies are working towards that. And in the near future, we will probably have a much better situation in this regard. And with this, I want to close my presentation and thank you for listening.
Hi, everyone. My name is Zhi Xie. I'm the Chief Medical Officer in Embed Therapeutics, and it's my pleasure here to present the topic decoding ctDNA MRD, how this kind of methodology can revolutionize clinical trials designs and to accelerate drug development. So my talk will divide it in two parts. The first part, it will talk about the value of ctDNA MRD and how it could facilitate the clinical trial studies. And then the second part, we'll talk about the current achievement and the outlook of this in precision medicine development. So let's go to the first part, the value of ctDNA in clinical studies. So the ctDNA in clinical study, the potential use can separate into two parts. The first one is that we can use the ctDNA MRD for patient enrichment, and then we can also use this ctDNA MRD for to serve as an alternative surrogate endpoint. So the first one is the patient enrichment, and the strategy use this the ctDNA MRD in clinical trial. Primarily, there are three strategies. The first one is that we can use the ctDNA to serve as a patient stratifications. So here we take early stage solid tumors, for examples, that before and after the neoadjuvant therapy and surgery, we can take a baseline ctDNA and take a post-surgery ctDNA, and then we stratify those patients into two groups, the ctDNA positive groups and negative groups, and then we randomize both groups, the patient in both groups to standard of care or standard of care plus investigational drugs. And then the endpoint could be conventional one, which can be disease-free survival or overall survival. And then we can also take serial ctDNA for exploratory. So in this way, we enroll all patients, but we stratify it based on the positivity of ctDNA. This is the first strategy. The second strategy is, which is called an enrichment strategy, that we only enroll patients with positive ctDNA and randomize those patients. And because those patients are considered to be higher risk, so we can anticipate a higher event rate, and we can allow a smaller sample size and also possibly a shorter trial time frame. And the third strategy is the adaptive one. So the trial design will be very similar to the first strategy, but we can do an interim analysis to see if there is the additions of investigational drug can actually have clinical benefits in which groups. So if there is the fertility test failed, then we can drop that cohort. So in that, we can actually make this trial more efficient, and we can close the cohort if that is there is fertility. So let's take one study, for example, which is IMvigor010 study. So this study enrolled high-risk muscle-invasive urothelial carcinoma, and then those patients were underwent radical surgeries and then randomized to receive adjuvant atezolizumab or observation only. Disease-free survival is the primary endpoint, and unfortunately, the primary endpoint did not meet. This is a failed studies to demonstrate the additions of atezolizumab have clinical benefits. But the ad hoc analysis showing that if we separate those patients based on the ctDNA positivity after surgery, you can see that the ctDNA positive patients actually have a worse overall survival compared with ctDNA negative ones. And then the additions of atezolizumab actually can have more benefit in the ctDNA positive populations. So this analysis actually lead to the designs of IMvigor011 study on the right-hand side. You can see that with similar patient enrollment criteria, but the study only enrolled patients with a ctDNA positive within 21 months after the cystectomy, and then randomized in 2-to-1 patients to receive atezolizumab or placebo. So we look forward to see the result of this study. We hope that that will be a positive result. Another example is a hypothetical one, which is an article published in JCO Precision Oncology 2 years ago. The article hypothesized that there's a study to investigate the patients with stage 3 colorectal cancer after curative surgery, and then to determine if the enrichment for ctDNA positive and can actually result in a more efficient trials. So the study design is that if we incorporate the ctDNA positivity in the patient enrollment and then randomize those patients to receive standard of care or standard of care plus investigation drugs, then the result, the simulated results is the table on the right hand. You can see that when we use the ctDNA to enrich for enrollments, the percentage of patients can be enrolled is only 19% versus the 100% with all-comer strategy. But the total screen patients number actually dropped from 1,600 to 1,000. And then the sample size dramatically dropped from 1,600 to roughly 200. So the sample size reduction actually reflects the reduced total cost of these trials. So in this article, it's clearly that the additions of the ctDNA enrichment actually can reduce the sample size and also reduce the total cost at the same time. Another use of ctDNA MRD in clinical studies is to use it as an alternative surrogate endpoint. So in clinical studies, we always want to know the treatment outcome, the treatment whether or not can lead to a final clinical outcome. And the clinical outcome in clinical oncology study usually is PFS or overall survival. But this kind of clinical outcome needs a lot of time to observe. So and surrogate endpoints that can be an early indicator of clinical benefit is always very welcome. And in oncology, objective response rate is often used as the surrogate endpoints. But are there any other surrogate endpoint we can use? So in 2013, the FDA approved the use of pathological complete remissions for accelerated approval in the neoadjuvant settings for high-risk early breast cancer. So this kind of pathological complete remission actually can be associated with a better long-term outcome. So that's the reason why we can FDA recommend to use this endpoint to serve as the surrogate endpoints. And in the I-SPICE 2 trials, as a retrospective analysis, showing that the pathological clinical complete response actually can be predicted by the ctDNA MRD results, and also the ctDNA MRD result also can be associated with improved survival. So in the future, we might consider to use the ctDNA MRD to serve as the surrogate endpoint in this clinical setting. But more clinical data are required to support this kind of applications in the future. So to summarize that, the ctDNA MRD analysis actually can help us in clinical studies to enrich for patients with higher risk, and therefore can reduce the number of participants needed, and also potentially cost savings for clinical studies. And then it can also be used as the surrogate endpoint, so that we can have a shorter follow-up times and try to accelerate the drug development using this kind of surrogate endpoints. So here we show some examples of clinical trial using ctDNA MRD. So you can see that some trials will use ctDNA MRD for patient enrichments, and it is included in the inclusion criteria. All those studies are in early breast cancer and only recruit patients with positive ctDNA. And then some studies, the BESPOKE study use the relapse of ctDNA MRD as the primary endpoint, which is also known as a molecular relapse as the early endpoints. And then some studies use MRD as patient stratification factors. The second part of my talk would talk about the current achievements and outlook of ctDNA MRD in precision medicine. So the first one I would like to highlight is that earlier this year in April, FDA ODAC actually voted MRD as the surrogate endpoint in multiple myeloma studies. So it's a historical moment because it is the first time that MRD-based endpoint has been recommended by FDA to be used for regulatory approval. So this decision is based on 20 studies, more than 12,000 patients' data, that we can see that using the log 5 log reductions as the endpoint, and you can see that on the right-hand side that the PFS of those patients can be clearly separated into two groups. The MRD negativity actually can predict the longer PFS, regardless of the subgroups, such as including newly diagnosed multiple myeloma, whether or not it is the patient are transplant eligible or ineligible, or in relapsed and refractory multiple myeloma. The positive MRD can predict worse PFS outcomes. And this is very important for multiple myeloma because nowadays the PFS for multiple myeloma is more than 6 years, and the overall survival, the median overall survival are more than 10 years. So a shorter surrogate endpoints that can an earlier endpoint to surrogate the treatment effects is really important for future drug development. FDA actually announced the guidance for industry that to use circulating tumor DNA for early stage solid tumors development, and 2 years ago. And according to that guidance, I think for solid tumors, further data are still required to support the use of ctDNA as the endpoint. And then, of course, the we just know that earlier this year that multiple myeloma has been declared that MRD can be used, but for all other solid tumors, I think more data are still needed. And there are challenges and limitations of ctDNA MRD to use to support regulatory approval. I think the first one is that there are multiple MRD technology on the market nowadays, and the performance and the platform differs. So the performance and standard is not standardized. So that that are major hurdles to let us to do a cross-trial comparison or to aggregate all the clinical data to support the evidence to be used as a surrogate endpoints. So that I think there's a major challenge that we need to some standardizations of all those MRD technologies. And then the optimal timing for sample collection, storage, and processing is not standardized yet. And of course, the shedding of ctDNA dynamics also varies by from tumors to tumor types. So and even from different stages. So that we still need more knowledge and more data to let us know the optimal timings for sample collections and storage. So here are my key takeaways. That the first one is that incorporating ctDNA MRD in clinical study designs actually can help us to enrich for patients enrollment that can allow us to have a smaller sample size and more efficient trials. And also the incorporate this kind of early endpoints as a surrogate endpoint and can facilitate the drug development. The second one is that the use ctDNA MRD in clinical trial to support regulatory approval can be used in multiple myeloma, but for other tumor types we still require further data to support that. So therefore, I think the agency actually encouraged all our sponsors to include this kind of methodologies as an exploratory endpoint in clinical studies, so that we can generate more evidence to support the usefulness of this technology as a surrogate endpoint in all other tumor types. So here conclude my speech. Thank you.
Hello, everyone. Thanks, GenomeWeb, for providing us the opportunity to present. On behalf of Burning Rock, I'm going to present my talk today is exploring the role of MRD in oncology drug development. I'm the Chief Technology Officer of Burning Rock. Today, my topic will be more focused on technical side. So let me just briefly introduce who we are. So Burning Rock is an oncology diagnosis company in China. It's been founded about 10 years ago. Right now, we are a NASDAQ listed company, and we have a CLIA and a CAP lab both in China and the US. And we've been working on different angle on focusing on oncology, but working on different kind of angle, including the therapy selection, early detection, and MRD. We're also doing pharma research service. Today, my topic will be focused in 3 parts. The first part will be talking about technical challenge of liquid biopsy and MRD. Second part will be the tumor-informed and tumor-agnostic MRD developed in Burning Rock. Some introduction on this. The third will be the developing platform we call Spiral. This is a method for a single portion of the ctDNA input to get multiple biomarkers profiled simultaneously. The technical challenge of liquid biopsy and MRD, I think everyone's been seeing this review paper graphic multiple times. The major take home here is very low, extremely low ctDNA abundance observed in patient after reaching from the surgery. Estimate about like the median ctDNA abundance is about 0.01% in the patient. So in order to achieve the MRD detection, we have to overcome this low abundance. So low abundance, which means the sensitivity is a major bottleneck for the MRD test. So how to get about 0.01 to 0.05% out of like a 10,000 DNA fragment, find needle in a haystack. So we have to find a very specific sensitive assay to perform this MRD test. So in order to achieve the signal detection, we also need to reduce the noise. So that's why the specificity is another challenge to achieve the effective detection. So there are two types of noise, the technical noise and the biological noise. So first, technical noise coming from DNA extraction, DNA degradation, also the sample preparation, library preparation, PCR amplification error, and also the sequencing error. It also comes from the post-processing error, which means coming from the bioinformatics alignment, all those things. So we are trying our best trying to overcome this technical. The biological noise coming from the non-disease mutation, not coming from cancer, that's such as the CH mutation due to age. Also the DNA methylation, another very high biological noise. Older people will have a more loci methylated. So how to differentiate this from the patient? So in order to improve the MRD sensitivity and specificity, I think Dr. Nitin is so including the improve the assay performance, reduce the noise, as well as increasing site detection, not only looking for a single site or oligo site, need to looking for multiple sites. So so my second topic will be talking about the tumor-informed and tumor-agnostic MRD assay developed in Burning Rock. As you can see here, the tumor-informed MRD is very important, very popular assay in this field. So there's but even for tumor-informed, there's a off-the-shelf panel, like a fixed panel, or to profile the tumor, or you're using a personalized panel to design based on the tumor profiling to design a patient-specific individual panel for MRD detection. So the benefits, the pros and the cons of both assays being listed here. So as you can see here, even using WGS, you can still think about this off-the-shelf panel, which means you are using WGS to profile the tumors and using WGS to profile the plasma for MRD detection. But for the tumor-informed one, like Burning Rock's PR-Profit assay has been launched about 2 years ago. We are using a personalized assay to design the MRD panel from whole exome. For both of assay, tumor-informed definitely give us a lot of advantage in terms of the sensitivity and specificity. For sensitivity, you can lower the cutoff. The reason is you already know what mutation you are looking at it. And specificity, so basically, due to you are using the personalized tumor-specific mutation, that's why you can avoid this biological noise. Using the PR-Profit, like I mentioned here, we perform the metal assay. The PR-Profit briefly is using the whole exome sequencing to looking for up to 50 somatic tumor-specific mutation, and using a personalized panel to do a deep, ultra-deep plasma cell-free sequencing, and using the Profit algorithm to perform the MRD coding and estimate the ctDNA fraction. And by using this technology, we are working with physician and published non-small cell lung cancer cohort in Cancer Cell last year. So briefly, the metal study I mentioned here is enroll about like 181 non-small cell lung cancer patients, mostly will be early stage. And we did a landmark and as well as a longitudinal study and demonstrated the efficacy of the PR-Profit assay, which is useful for MRD detection. Today, I'm going to switch the gear a little bit about discussing the technical consideration. So there's 4 different kind of technical aspects when we do the tumor profiling, and on the using the tissue sequencing, what kind of a consideration we need to looking at, what kind of a tissue panel we want to use, and also the biomarker type and the mutation type of selection, as well as some technical design consideration. So the first slide talking about the tumor profiling, when we look at the panel size and the tracking mutation number, as you see here, The more tracking site you're looking at it based on this in silico model. If you select the 50 loci for the selection on a routine like a 20 nanogram input of a cell-free DNA, you can reach about 4.05, actually 5 LOD. So this is actually what BR Profits can achieve. So by doing this, you will definitely overcome, like say you only have a very few loci. One thing I want to point out, if you're using like a regular around 500 gene panel due to tumor profiling, so the loci number you usually can get based on different cancer type, you will get a different kind of number of the mutation, meet about 10 loci. But if you talk about that you need 50 mutations out of this 500 gene panel, very few cancer type can achieve those, and very few individuals. I think this is things you need to consider. The bigger panel, larger panel usually give you a better choice. The tumor profiling, the other part will be the biomarker type. Single nucleotide variants is the most popular one, but even for single nucleotide variants, if you can do phased variants like what Foresight Genomics doing, it can improve the specificity. The reason is it can reduce the technical noise dramatically in the copy number as a tumor-informed calling. So like C2I Genomics demonstrate like when they do the tumor sequence first to get the CNV information, then do the plasma sequencing, get the CNV information based on the tumor CNV, you can still much improve your assay, sensitivity. So the other aspect will be the mutation detection. In METAL study, we're looking at canonic driver mutation, driver mutation EGFR. So even for the non-canonical mutation MRD positive, the survival curve still will be much worse than the MRD negative patient, which means the non-canonical MRD mutation type is still important. So you are not necessarily always just looking at the driver gene. So the fourth consideration will be more technical, that related to the design. So as you can see here, when you tumor profile, when you sequencing the tumor, you will probably get a lot of a mutation if you do whole exome sequencing. What kind of a mutation will be good for MRD personalized design, or which was good for MRD tracking? So it depends. Like you need to rely on a lot of technical detail. For example, on the left page, we see the good alignment picture on the sequencing, and on the right part will be the bad alignment. You can see a lot of soft libraries. So even they probably both could be the true tumor mutation from the tumor perspective, but only the left one could have potential for MRD detection. Since if you're sequencing on the cell-free DNA, you need a very low noise. So any kind of a noise can basically make that site ineffective. So I list a couple of those different kind of aspects. I don't want to read them through. You probably can take a look at basically the experience we achieved, or kind of when you design the tumor specific panel, what kind of aspect when you're talking about it. So that's the summary here. I don't want just to go through this again. And so the other thing we want to take a look is the methylation. So methylation on the other side will be totally different angle compared to SNV. So methylation, the key point here, the signal recurrence rate is very high compared to single nucleotide variation tracking, the somatic side. So you can see on the right page, the highly recurrent marker across different kind of a cancer type, they all showing very recurrent signal here. So that's why it gives us the opportunity to tumor diagnostic panel looking at the methylation. In terms of methylation as a tumor agnostic, so which means we based on the TCGA database, our internal sequencing result, as well as the plasma sequencing result, we refine initial panel for this study. We did a sensitivity assessment by doing the cell line contrived signal and dilute down to 0.004% with 40 ppm. As you can see here, even at the tumor agnostic, without the tumor prior tumor information, we still be able to detect down to like 10 to minus 4%, 10 to minus 5 level of LOD. And when we do the specificity check, we actually using BR profit, which is a tumor-informed negative cell-free DNA as a negative, we think it's a true negative cohort. We tested about like more than 300 cell-free DNA sample, and you can see here almost everyone being all negative. Specificity still can be held without like knowing those kind of information like prior. So very lastly, I'm gonna introduce the Spiral platform, which is we currently developing, and there's a method for a single portion of a seed to get a multiple biomarkers profile. As you can see here in this field, there's a lot of companies been trying doing this. For example, Guardant launched their Guardant Infinity assay for biomarker biopharma service. They're looking for epigenetic genetics in the same time. And the biomodel also published a paper launch product for 5-letter sequencing as well as 5MC methylation. Internally, we developed a similar program actually in our internal proprietary technology, trying to utilize a single portion cell-free DNA to serve the purpose, get the both methylation signal as well as any kind of a somatic signal, even in the fragmentomics signal if you want to take a look. So in order to demonstrate the efficacy of this technology, we actually do a ctDNA cell-free DNA contrived sample demonstration to demonstrate the POC. We're using 10 nanogram as input, and we create the spiral libraries and cover two different kind of angle. We look at BR profit, we do the LP low pass WGS. We also do on-whole panel sequencing. More importantly, we do the methylation panel like I mentioned earlier. Do the tumor agnostic. We see here on the top panel, we are using the classic BR profit about like 40 ppm LOD sensitivity, which is on expectation. On the bottom part, we're using the methylation panel in developing. We can achieve about like 125 ppm, which is about a 2 to 3-fold compared to a tumor-informed panel. But that's still very promising. The reason is that this is tumor agnostic. We don't know the tumor methylation information. We just need to look at the plasma only. More importantly, as you can see here, for the contrived diluent, we got 100% DNA using this methylation, which means it's the specificity can be secure. Of course, we can look at the public number variation. The proof of concept study rely on the tumor sequencing to get the CNV information. Then we do the serial dilution using the same assay, same library. We sequence whole genome sequencing, and the tumor-informed the CNA score software can reach about LOD 0.1%. But this is still significantly higher than the tumor-informed BR profit as well as the tumor-agnostic methylation. Overall, in conclusion, so I think how to overcome the ultra-low signal abundance will be important for the MRD detection success. And the Burning Rock actually has both tumor-informed BR profit assay to demonstrate the 40 ppm LOD. And meanwhile, we also develop a methylation-based tumor-agnostic assay to hopefully we can reach this sensitivity in our final product release. But it's still on the way. Of course, we developed another thing. We developed a spiral technology, which is using the single portion of the cell-free DNA to profile both genetics and epigenetics marker. So this is actually providing new opportunities for biomarker study to serve serve our pharma partner in the drug development and the translation of the study. This concludes my slide. Thank you, everyone.
Great, thank you, Joe. As a reminder to our webinar participants, if you have a question, you can type it into the Q&A box in the control panel. We'd like to ask attendees to take a moment after the webinar has ended to take our exit survey and give us your feedback. We will now start the Q&A portion of the webinar. The first question is for Johannes. As tumor-informed MRD has gained increased usage recently, what do you think will be the opportunities and challenges for adoption of tumor-naive MRD in clinical studies and clinical practice?
Yeah, I think there might be a reasonable difference between clinical study and finally clinical practice and where it is done. I feel that like an individual, personalized, informed ctDNA panel is probably the most sensitive for the tumors and also for the patients. And actually in this Galaxy CRC study that I presented, where we really had this very high predictive and prognostic value of the ctDNA MRD, this was actually used. I am wondering if this might be too complicated for non-university or non-first-tier clinics in the end for the clinical practice, or like in my home country in Germany, where many oncology patients are treated in outpatient clinics, in oncology practices. I'm not sure if this currently is feasible, although I have to say that besides the fact that Joe just presented with methylation assays and some other topics where I am feeling that maybe there's really a way to distinguish tumor cells from non-tumor cells, not only based on the particular tumor or the particular pattern in the tumor, in the individual tumor, or at least in the indication. So let's say before that, I was feeling that tumor-naive assays might not be the right place for MRD assessment, but maybe there's a chance. Maybe the providers of the panels can, in the end, also find a way, because this would make things much easier for practitioners, for clinics, for patients. I don't think we are there yet.
And is there a role for companion NGS and digital PCR panels for MRD?
No, companion diagnostics is kind of a difficult topic for me, because I mean we just finished the area of for example all the PD-1 inhibitors, where everyone had basically with the same target a different companion diagnostic. And so if we have now, as also the speakers before me said, if we have like 100 different panels that compete for doing this, and then one company develops a new compound going with one panel, another company goes with another panel, and in the end we have this big variety. I don't think this is good for the clinical practice in the end. So I hope that authorities will come to a way where not the particular panel, but the principle can be approved, and this will make things easier for us.
Chi, what do you think is the popularity of using MRD with patients these days? Are physicians accepting it, and will it be a standardized solution in patient treatment?
Yeah, I think thanks for the questions. I think it's really important. So I would say it's based on which tumors are you talking about. So for leukemia, for the especially chronic myeloid leukemia, physicians already adopt the MRD testing for years. So for leukemia, such as acute leukemia or multiple myelomas, a lot of physicians already adopt MRD during the day-to-day practice. But if we are talking about solid tumors, it is completely different. I think the popularity of using MRD in solid tumors is still very limited. I would say most physicians did not accept the idea to use that. But nowadays, just like I mentioned, that FDA actually encouraged sponsors to use MRD in solid tumors, and then a lot of symposium and a lot of discussion sessions in major conferences such as ASCO, ASC, all talk about the utilizations of MRD in the management of patients even in solid tumors. So I anticipate that more and more physicians will accept this idea.
And how might using ctDNA MRD for patient selection impact patients' accessibility to clinical trials?
Right. I think to utilize the ctDNA MRD for patient selections, I think it can be more precise. So because we focus on the high-risk groups, high-risk patients, which could suffer from early relapse or early progressions. So those patients will benefit the most from the investigational drugs. But using this technology, of course, they will limit the patient's accessibility to clinical studies, because if a patient has MRD negative, then the patient cannot be enrolled in that study. But I think I'll see this from a positive perspective, because we are selecting more precise patient populations who can benefit from the treatment. So I think it is not only more efficient from sponsor point of view, but it's also in the best interest from a patient perspective. So even it will impact the accessibility, I think it's still positive in the long run. If more and more clinical study using ctDNA MRD as one of the patient selections, then I think most patients can still be enrolled based on based on the best interest of their own.
Joe, what difficulties would you consider when validating an MRD solution for clinical practice?
I think for clinical practice, I think trying to get the clinical validity and the utility will take much longer. MRD, the reason is take multiple years to get clinical information out of that. So the other things about like disease of complication of disease, different kind of indication, and all those things actually bring a lot of also the difficulty of the statistics itself, trying to how to design a good clinical trial, and all those things will be provide additional challenge for MRD.
And what are the greatest challenges with MRD panel design? And what are the turnaround time expectations for MRD test results?
For panel design will be like more technical questions. So how do you differentiate what kind of mutation, even for tumor-informed, what kind of mutation you will be used for tracking, and whether it's being reliably detected, and whether this mutation is a stem mutation, which is every tumor cell contains, and or it's a branch mutation, or only a subpopulation. All those questions will be important for effective MRD design. And the turnaround time expectation, I would just say, for tumor-informed, we expect like 4 to 6 weeks. Better will be 4 weeks from the tissue biopsy till like first MRD data out. But this is actually very challenging. Reason you need to customize and need very good logistics. The other part for plasma OLE or tumor agnostic assay will be just as a routine test. So it takes only about 1 to 2 weeks expected.
This next question is for the three of you, and Johannes, you spoke to this a little bit already. What do you each think of tumor-naive versus tumor-informed approaches to MRD? Are there different settings where one might be more advantageous than the other?
I mean, it depends on, as I said, it depends on the availability. It depends on the level of the hospitals that we are doing this. The most advanced and the most desired for me is still the patient-specific informed one. And but I mean, for example, what we could think of if this tumor-naive ones are easier to do, maybe even less costly in the end, because you don't have to do the tissue analysis or so. It could be, for example, that if they are quite sensitive, that doing a naive panel first with a high sensitivity, and then based on the result, if the result is showing that there is ctDNA that might be a tumor in the end, or might be a minimal residual disease, that you can then send the patients to a hospital or do something that these things can be done and can be easier done. That would, for example, be an approach to start with a simple and continue with a complicated. But on the other side, if you don't take the tumor sample and test the tumor sample directly after the surgery, the results also of the informed one will be less there. So I think if we are good on the way of developing the tumor naive ones, that they are reaching the sensitivity, the predictive value also of the tumor informed ones, I think that would probably be easier, because this kind of a one-in-all solution is usually what I prefer. But in the times of precision medicine, where we are also tailoring the medications to the particular patient's tumor, like we are doing in many cases also, I think also it's worthwhile to continue with going further the same way with the diagnostics. Thanks.
Chi, do you have anything to add to that?
Yeah, I also agree that I think it depends on, it differs from tumors to tumors. So I think for prevalent tumors, or for tumors that can the tumor naive ways can have good sensitivity and specificity. Of course, the tumor naive ways approaches is always more welcome, because the shorter turnaround time and we don't need to have sufficient tumor samples from the patients. I think that would be much more welcome if we can achieve an acceptable sensitivity and specificity using this kind of approach. But I doubt that every tumor types will be able to use that. So in some cases, I think the tumor-informed ways still needed to have a better sensitivity and specificity.
And Joe, anything else to add?
I think I agree on both of you's assessments. So from technical perspective, I think right now we've been actually able both platform. I think it just depends. I hope like both application can be, serve as a comparator to against another one, and in this kind of we can find the utilization of both assay on certain kind of angle. So that's my assessment. So from a development perspective, I think we've been trying doing both, especially utilizing the technology we developed. We will try to find an answer doing this kind of a clinical trial or clinical study.
Well, that is all the time we have for today. We'd like to thank Johannes Nipkin, Chi Shea, and Joe Zhang, and our sponsor, Burning Rock. If we didn't have time to get to your question, we will provide them to the sponsor to follow up. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of the webinar, or if you'd like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us.